This is Design Safe Radio, where natural hazards researchers strive to make our society more resilient to everything nature throws at us.
So we're talking about the evolution of trees and microbes and all these things. Essentially, it's like shortcutting the engineering design process because it's already been done for you. It's now just kind of picking up where those evolutionary processes have left off and seeing, well, why did they, these various ways of growing tree roots, for example, why was that selected? Why are they optimal for a given soil chemistry, soil type, particle size, siting, temperature, all the things that go into making a tree root, and then applying that to things like, wind farms or other structures.
Yeah, exactly. That's exactly it. In evolution, in a way, has tried, thousands and thousands of prototypes for us. And so if we can understand and do a little bit of reverse engineering for why a given plant or animal looks the way it does, then that helps us a lot. And another thing that's really nice in these solutions is that animals and plants cannot use different materials that they have, right? They cannot use metals, for example. They only have a finite amount of energy. So in a way, a bio-inspired solution or biological solution needs to be energy and material efficient, almost by definition, because if they need more energy, they cannot necessarily just find more energy. They cannot just fill the gas tank with more and keep going. They need to hunt, they need to do all these things. And so they also tend to be very efficient energetically and in terms of material usage.
Yeah. Have you collaborated at all with the University of Arizona, the Biosphere 2 team over there that's doing a lot of studies on plants in different biospheres?
Not with them. I've never collaborated with them, but we did collaborate with plant biologists at Davis. We're an ag school in Davis, and so when we started this research on the root-inspired foundations, we certainly worked with biologists, and we actually did some field tests on trees, and so we literally tested trees as sort of anchorage systems, and we pulled them out of the ground with the purpose of trying to understand the relationship between the uplift capacity of a root system and the architecture of the roots. And what it looks like.
Wow, that's really cool. I think I remember that test. We'll have to find a way to link to the data from that test for anybody who's interested and
Sure.
put it in show notes. So kind of speaking of Davis and everything that's going on down there, you guys are affiliated with this another National Science Foundation funded center for Bio-mediated and Bio-inspired Geotechnics, CBBG. It's even hard to say the acronym for that. So can you tell a little bit about these centers, how you're related, and how CGM is a test bed for those CBBG ideas and some of this emerging technology?
Yeah, of course. The CBBG is an engineering research center funded by NSF, and so essentially an ERC, that's the acronym. It has the goal of developing new technologies and then translating them to practice, to things in ways that will help society at the end of the day. And so this one focuses on geotechnics, but with the spin of bio in it. And so there's 4 universities that are part of it. Arizona State University is the lead university, and then Georgia Tech, New Mexico State University, and UC Davis. And so the interaction between the CBBG and the Center for Geotechnical Modeling, the CGM, is that the centrifuges are a test bed for the CBBG. Essentially, is an equipment site that provides capabilities to help in this sort of translation or upscaling of the technology from the laboratory, right, where it's very fundamental, all the way to the field where we can actually deal with all the complexities, like the effects of installation, and how it'll behave when it's, really a really big foundation. It's going deep into the ground for tens of meters and things like that. The centrifuge sort of fills the gap in between those two skills. And so, yes, there's been a lot of interactions. It's used for CBBG projects pretty regularly. I use it in both projects that we have active with the CBBG right now. One project deals with piles and anchors inspired by snakeskin. And so we look at this really cool property of snakeskin, where if you rub it in one direction, is very smooth, but if you rub it in the opposite direction, is really rough. And so in biology, they call this frictional anisotropy or frictional directionality. Essentially, the amount of friction depends on the direction of movement. And so we're designing surfaces that are inspired by snakeskin that can do the same. And then we're putting them on piles, and then we can test the piles in the centrifuge, for example. Another one deals with site characterization tools that we use to determine soil properties, but that are inspired by other animals like earthworms and plant roots and things like that, with the goal of being able to characterize sites that are very difficult to access. For example, underneath an existing dam, it's near impossible if we have uncertainty on what the soil conditions are down there, right? It's near impossible to take samples and do tests on that soil. Similarly, underneath existing structures, it's pretty difficult to test those soils. And so we're developing that kind of technology, and the centrifuge is really helping there, because one of the things that are really important, and I'm sort of always talking about this, is in bio-inspired research, we do need to be really careful in addressing the difference in depth of what we see in nature to what we're gonna ultimately apply to in geotechnical engineering. And that's because if you think about it, tree roots, earthworms, moles, whatever you want, all organisms pretty much live in the top 5 feet of the ground.
Right.
And the pressures and the stresses and all of that is very small compared to the stresses that you get in the soil, 30 meters deep. And so that's actually a pretty big difference, and we need to take that into consideration. And so the centrifuge really sort of fills that gap, right? In the centrifuge, we applied this increased gravity that allows us to model that larger depth, and that's relevant to geotechnical engineering. So those are two examples. Other examples related to the biocementation that I was mentioning earlier. For example, Jason DeYoung, who you mentioned had another episode earlier, they're looking at how biocementation affects the liquefaction behavior of loose sands, right? West Coast, it's very seismically active. I mean, other areas in the US as well. But if we have sands that are relatively loose, those can liquefy, lose their strength. And so it's pretty difficult to treat an entire site and wait for an earthquake to come to see how it's going to behave. An easier way to do this research is to do the treatment and then shake the ground, and we can do that in the centrifuge. And so, we've been done that. They have been doing that. A team from Arizona, ASU, a couple of years ago, also did a similar test, but with the bio desaturation technology as well. And so they also saw pretty promising results.
I think they've also done some field tests collaborating with the UT Austin shaker trucks, if I remember right.
Yes, they certainly did. Yes, if I remember correctly, they did the centrifuge work first, in a way to, like a proof of concept. Is it going to work in conditions closer to the field? And then based on that, and things worked well, then moving to bigger scales. Now using the Texas shakers out in the field.
Yeah, we continually on these interviews just get to show off how helpful it is to have this network of facilities supported by the National Science Foundation, right? We have the ability to do smaller scale on the 1 meter and 9 meter centrifuges. And then once that proof of concept is developed, then you can go out into the field and have the Mung and the shaker trucks out there, and really see what differences there are in real life when you have other soil types and other factors like weather and all the other things that go on out in the field.
Thanks for listening to today's episode of Design Safe Radio. Be sure to like and subscribe on whatever platform you happen to be listening to this on. It really helps people find our show. Thanks to our amazing sponsors, the National Science Foundation and the NHERI Network Coordination Office, which is award number 2129782. Big thank you to Marty LaChance, our guest booker and topic researcher extraordinaire, and Raquel Ruiz, who is our video and audio editor. I'm your host and NHERI Facility Scheduling and Operations Coordinator, Dan Zahner. We'll see you in the next episode. Until then, stay resilient.